ALS is the most frequent adult-onset motor neurodegenerative disease. It results in paralysis and eventually death due to respiratory failure. ALS is characterized by selective degeneration of motor neurons. Nevertheless, cell types other than motor neurons have been implicated in ALS pathogenesis. However, it is still quite controversial whether or not also the skeletal muscles are contributing to the disease. Our lab showed that the skeletal muscle is actually playing a more active role in the degeneration of neuromuscular junctions, which is an early event in ALS progression.
What this voucher will allow us to do is actually to join our expertise between the Donders Institute and the Rimmels at the Radboud University. And so using our mouse models, we can actually dissect the contribution of the skeletal muscle and the presynaptic nerve terminal at the neuromuscular junction in the pathology of ALS. So what we want to do is to dissect this neuromuscular junction, look in particular at the nuclei that are just below the synaptic cleft, which are supposedly more affected by the disease, and also look at what's the impact of the disease on the presynaptic terminal side.
After these samples have been prepared and the nuclei isolated, the people from the Donders will run across the street to the Rimmels, where we will take these samples and profile at a single nucleus level all of the genes expressed. This will allow us to look at differences in the subsynaptic nuclei versus all the other nuclei in the wild type and the ALS model mice. At the same time, we'll also be expression profiling the nerve termini from the same samples to allow us to, in the end, tease apart the effects of the nerve on the muscle and of the muscle on the nerve.
This project is very timely. We are bringing together disease modeling and single-cell genomics to understand the cellular basis of disease. And we thank the university for providing this initiative, and we have the expectation that this will lead to further collaborations in the future.